Hi, my name is Daniel J. Gómez, and I just wanted to give some upgrades on CRISPR following the talk that I'm giving today in Biology 622 Advanced Molecular Techniques. So I'm giving a talk on CRISPR-Cas12a target binding unleashes discriminant single-stranded DNA activity, and this is by Chen et al. And Jennifer Doudna's group at UC Berkeley, as well as the College of Chemistry and the School of Medicine. So there's also other collaborators on this paper, which is the HHMI or Howard Hughes Medical Institute and Innovative Genomics Institute. So there's the Molecular Biophysics and Integrated Bioimaging, MBIB, as well as the UCSF School of Medicine on this paper. And it was really innovative paper for the time it came out was 2018, 2019, they were depositing data. And it's, again, about CRISPR-Cas12a. And I just wanted to speak to some of the current developments in the technology. Just to give you guys some background on the CRISPR-Cas system, they're RNA-guided nucleases that provide adaptive immune protection for bacteria and archaea against intruding genomic materials. So they consist of the Cas or CRISPR-associated protein and CRISPR RNA or CrRNA that recognizes and cleaves foreign DNA. So the programmable nature of CRISPR targeting mechanisms have enabled their adaptation as a powerful genome engineering tool, which is widely used. So the CRISPR-Cas9 complex that you see, this is a system that's the most widely used in genome editing as a genome editing tool. And it consists of the Cas9 protein and a single guided RNA that directs Cas9 to cleave specific DNA sequences. The system has revolutionized genome engineering, has been found in applications such as agriculture, medicine, and basic research. CRISPR-Cas12a system is a type 5 CRISPR effective system. So this is the other one in gray. And you can use this as a single RNA guided endonuclease. And that's why they call it Cas12a. And it's formerly been known as CPF1 to target and cleave DNA. Unlike Cas9, 12a generates a staggered cut in the target DNA, leaving a 5' overhang that can be used for DNA manipulation. And it's been found to have several advantages over Cas9. So these include having it's a smaller size, and then it has the ability to cleave DNA more efficiently with a single CRISPR RNA or CrRNA. Additionally, Cas9 or Cas12a can recognize, so the 12a can recognize and cleave a broader range of DNA sequences, which may increase its potential in applications for genome engineering as well as molecular diagnostics. Just showing you visually what's the difference between these two Cas proteins. You have the Cas9, which makes a blunted end cut at NGG, and the guide RNA is it's going to be 103 nucleotides. And then for CPF1 or Cas12a, what is known now as is a 5', you get a 5' overhang cut at TTTV, and that's 41 nucleotides. So you can see the difference not only in the size of the Cas protein, Cas12a protein size, but also its gRNA requires less. So it's 41 nucleotides, and it provides a 5' overhang cut, which could be more useful for molecular biologists. I wanted to show you guys the cryo-EM structures that I went over today in class for Cas12a. And this includes a single particle analysis cryo-EM of Cas12a. So you can kind of see it in its recognizable mouth that looks like it's coming this way. And you can kind of see the inside of where the endonuclease activity is. And the Q-score for this is 0.549. And the Q-score can go between 0 and 1. So you can see there's room for improvement here. And it's at 3.0 angstroms. So consider that. And then I'll just go ahead and show you the cartoon of Cas12a just to remind you guys what it looks like. So it has the CRISPR RNA, the dsDNA, double-stranded DNA target, as well as the PAM sequence in Cas12a. If it's double-stranded DNA, then it has to have that PAM sequence, which is TTTV. And then you have the RUVC or RUVC location as well, which facilitates the nucleus activity. And then as a strategy, there's this double nicking dimers, and we'll talk more about this later, but you can actually dimerize the Cas12a proteins to make for a more efficient and specific cut. So I'll go over the specific distance that is required, but you can see here they have a single particle cryo-EM structure with a Q score of 0.411 of dimerization of Cas12a. And then as a binary complex, meaning that the single guided RNA is attached to it as a system, you have this cryo-EM image at 3.2 angstroms using single particle analysis, where you can see the single guide RNA, the nucleic acid ribbons, as well as the protein characteristic alpha helices. So what's going on in current developments? We'll go over this dimerization as a double nicking strategy. So you can have two, in this instance, Suzuki in 2021 did Cas9, and he dimerized them with an offset of a guide RNA offset of 9 base pairs, meaning you can increase the specificity by doing that. And then this is the Zhang's work of getting the cryo-EM image of the same bacteria that we talked about today in class, LB Cas12a complex with the CRISPR RNA, as well as the Acer V8-4 dimer. So that's a dimerization at 3.38 angstroms with a Q score of 0.411. And that shows the dimerization of the Cas12a. So we have Cas9 in the cartoon, but Cas12a in the cryo-EM bioimage. So this double nicking strategy with Cas9 and Cas12a can benefit genomic or genome engineering and even gene writing in several ways. So you're using two different nucleases, which creates a pair of nicks in the DNA, and that the probability of the off-target effects are greatly reduced. So you can enable more precise genome editing, deleting, correcting, or inserting. So you're using two nucleases to enable more efficient editing. So the two nicks itself can stimulate more efficient DNA repair mechanisms like homology-directed repair. So that's HDR. So you introduce precise changes in the genome this way by using the double nicking strategy of the dimerization of either Cas9 or CPF1 or Cas12a. And then a follow-up study by Doudna as well as Ma et al., who was the first author, shows that you can actually engineer CRISPR-Cas12a to improve the genome editing. So this is taking an engineering approach instead of a dimerization. We're engineering the Cas12a as an RNP. So this is a ribonucleoprotein, which it is. You have the CRISPR RNA, and you can see the Cas complex with the Cas12a as a binary complex, and you can look at single-stranded DNA or double-stranded DNA as your target. And going for the double-stranded DNA, you would have to locate that PAM and have it to recognize the target, and then make a cis cleavage, and then you would activate that RNP. So that activation of the RNP would then go through the trans cleavage, and then you would get single-stranded DNA. If you already have single-stranded DNA as a target, then you go straight into the activation of the RNP, make your trans cleavage, and then you have your single-stranded DNA there. And then looking at the sort of the primary sequence of the Cas12a, CRISPR-Cas12a, you have the recognition lobe and the nuclease lobe. So you can see them in their associated colors. And then they're really focusing in on this figure on the bridge helix. So this bridge helix, you can see as a specific target of the Cas12a. And what they're doing here is they're making mutations of the bridge helix to see how it goes about this endonuclease activity. So they switched 890, 883, 880, and 881 all to alanine. Whatever it was before, they just made it alanine. And then they wanted to see which ones were hyper effective. So you can see some of the results. This is really to improve the genome editing tools by rational engineering and directed evolution to generate the variants of LB-Cas12a, which it really did, and the mutants exhibited enhanced gene editing and HCR efficiencies, and this was across multiple loci in mammalian cells. So this was accessioned, and this is all in the NCBI. So what they have here now is a hyper-effective LB-Cas12a proteins. So they designed the bridge helix mutants and with beneficial mutants, and then they got the hyperactive mutants. So the hyperactive mutants would be the mute 2B and 2C W, and then they reversed mutation of at the location of 884 from leucine to phenylalanine, and then you got the mute 2C WF. So again, the bridge helix is very important. So they showed through their results that the Cas12a bridge helix differentially affected the cis versus trans DNA cleavage. And then the directed evolution of the mutant LB-Cas12a improved the genome editing activities. And then again, the reverse mutations in the bridge helix yielded these hyper-effective Cas12a genome editors. And this kind of shows you how they went about the selection of the efficient editing process. So they use E. coli with a dual plasmid selection system where they had, their CRISPR RNA, their LB-Cas12a, as well as the CAM resistance gene. And then they're able to select through the CRISPR RNA targeting and the CCDB gene using the PAM TTTG. So they use PAM instead of V, they use a G here, a TTTG PAM. And again, the suicide genes that we're using in lab in biology 622, they're using those suicide genes in the that are downstream of the ampicillin or the AMP resistant gene, as well as the CAM resistant plates as well with arabinose. So you have these hyper effective mutants that are more efficient at genome engineering, and they're showing you how they went about this, and then the different domains that they're interested in. So the bridge helix domain, the nuclease domain, as well as the RUVC domain, the RUVC domain. So those are the ones that they focused on with Ma et al. In 2022 to show that the bridge helix itself is very important. So then they're interested in the structural prediction of the LB-Cas12a mutant proteins by AlphaFold. So they use AlphaFold 2, and they also had macromolecular crystallography structures to compare. And this is what they showed. So they have a closed and open conformation for the LB-Cas12a. And they have it with CRISPR RNA as well as with CRISPR RNA and DNA complex with each other in the open conformation, right? Because if it's open, then you can have the DNA in there as well. So that's the PDB ID 5XUS that I use that we'll look at more later. But here we have the wild type, which is in cyan blue, and that's the wild type in the AlphaFold prediction, which takes a closed conformation. All the mutants are adopting this open conformation. So the predicted structures show the 3 active variants, which are in yellow, and those were generated in this study by Ma. And then they predominantly, again, open. They were open in the open conformation, whereas the wild type, the Cas12a wild type, predominantly adopts a closed conformation, and they were able to validate this through AlphaFold 2. And then looking at the MX structures, again, the same one that I mentioned, you have the Cas12a with the CRISPR RNA and DNA complexed. And what I did was removed the protein and just looked at the CRISPR RNA and DNA only to give you an image of that. And then I put it back in. I put it back into the protein, and I was able to orient it to show you this characteristic mouth-like, structure of the LB-Cas12a, where you can see here in green highlighted of the bridge helix, which again is very important, that bridged helix. Giving you a closer view looking into the mouth, you can see to the left this bridge helix that really facilitates this endonuclease activity of the Cas12a. And what I did is I took the sequence of that and then predicted the bridge helix structure in CoLabFold, and that's the bridge helix by itself. So we're the dark blue means it's a PLDDT score that's very high. It's close to 100 here, and you can go to 0 for 100, and they're very confident in that structure of the bridge helix. And here's another one kind of at a diagonal angle of the bridge helix complexed in the LB-Cas12a. So here's another paper in 2020, 2 years before that last study, showing that the bridge helix arginines for Cas9 they play a critical role in the sensitivity of Cas9. So, and especially when it comes to mismatches. So this is a group that works with Doudna, Emmanuelle Charpentier, where she was able to look at the glutamine residue, the Q768, and then look very close at that bridge helix arginine residue that happens over here at 71, 69, 74, and 75 that all interact with the CRISPR RNA. So that was her group that was able to show that in 2020, and that was published in Nature Chemical Biology. The year before, we have same Cas9 bridge helix that modulates the targeted DNA cleavage. So that targeted DNA cleavage it tolerates these mismatches. So you have the CRISPR-Cas systems that are able to protect against the intruding DNA. And again, this Cas9 is a CRISPR effector. It's a type 2 CRISPR effector that's used for gene editing. So understanding these different domains, like you'll be able to understand what's going on with the DNA-induced changes that enable the development of the Cas9 variants. So these prolines in the residues, they're substituted in the bridge helix. And in this one, they used S. pyrogenes Cas9 to improve the targeted DNA selectivity. So you can see the bridge helix loops or BH loops that may contribute to the targeted DNA recognition as well. So here you have a targeted DNA strand, the single guide RNA seed, as well as the BH loop. And this one has a tracer RNA with a stem loop 1 in there as well. So this was another study that was done the year before in 2019, showing the role of bridge helix and Cas9 in its modulation features. So getting back to this CRISPR-Cas applications, like I was thinking about like conceptualizing an RNP device. So I wanted to start with the particle of RNP, and just this particle where you can see the alpha helices and the antiparallel beta sheets, and the alpha helices are alternating in this PDB structure. And what do we really know about this? So we have a Cas12a, right? A type 5 CRISPR-Cas RNP that can be programmed to target and cleave specific RNA molecules. So when Cas12a is complex with guide RNA, it can bind to and complementary RNA sequences and induce cleavage to target RNA. So Cas12a can exhibit collateral cleavage activity, meaning that it can it can cleave multiple RNA molecules in a non-specific manner. So it's non-specific after binding that single target RNA, but maybe the dimerization that we showed, that maybe being able to do the dimer with the offset of 9 base pairs, you'll be able to adjust for that. So I also wanted to consider Cas-Rx, which has been recently discovered and characterized, which is a type 6 RNP, CRISPR-Cas RNP. And it's very similar to Cas12a, but it can be programmed with a guide RNA to cleave specific RNA molecules. So the specificity of Cas-Rx has some unique properties, making it a promising tool for RNA manipulation. So it can cleave single-stranded RNA molecules, which expands on the targeted capabilities beyond double-stranded RNA targets of CRISPR-Cas12a. So CRISPR-Cas12a can target double-stranded RNA, but this one can actually, Cas-Rx can target and cleave single-stranded RNA molecules. So it also does not exhibit the same collateral cleavage activity, which would reduce offset targeted effects. So how would we load this RNP into a deliverable? There's a microfluidic device developed by Suzuki in 2021, where you can take the Cas protein with the CRISPR RNA and tracer RNA and really complex it in the microfluidic device to load a lipid nanoparticle. And then Suzuki also provided electron micrographs, 10 images of the RNP loaded lipid nanoparticles, or LNPs. So the steps towards development here for an RNP device would be using Cas12a or Cas-Rx, which would involve complexing the RNP with a guide RNA designed to target specific RNA sequences of interest. And then you can target the RNA, and then the guide RNA would direct the RNP to cleave RNA at specific sites, leading to degradation. So the device can be used for a variety of RNA manipulation applications, including gene regulation, or RNA editing, as well as disease diagnosis and treatment, which is all really good stuff. So there's even room for growth further. So we can do now advanced nanotheranostics of CRISPR-Cas, and this was shown by Kong. So here we have the detector, which I talked about today in class, and which takes the non-specific DNA and the targeted with an RPA, as well as the DNA. And then you get a fluorescently quenched F reporter with the Cas12a. There's also other methods such as Sherlock and Sherlock version 2, CRISPR chip, CRISPR-Da, which all can take cells, blood, septum, urine, and feces and you have CRISPR-Cas detection. So with the CRISPR-Cas detection, you can detect viruses, cancer-related mutations, cancer screening, patient genotyping, as well as antibiotic resistance. And what's emerging from that, so we just reviewed, some of the advances in CRISPR-Cas detection systems. Now, what's emerging and potential for CRISPR-Cas is liver delivery via viral and non-viral systems. So you can see that figure here. And there are three formats of CRISPR-Cas systems. You have plasmid DNA, Cas mRNA, gDNA, and an RNP. So they can be packaged into delivery vectors for in vitro, in vivo, and further human clinical applications of liver-targeted gene therapy. And then we have integration of CAR T therapy and CRISPR-Cas technology. So in here we have combined CAR T and CRISPR therapy. So we can take an isolated T cells from the patient, that's here in step 1, and then do CRISPR-mediated engineering in the T cells. And then step 3, we amplify the edited T cells. And then step 4, we re-infuse the edited T cells into the patient. And then here on in this last figure is the rational designs of CRISPR therapeutics with cargos and delivery systems. So these are really what the better CRISPR therapeutics can be improved by both well-designed CRISPR-Cas cargos and safe, efficient, and targeted delivery vehicles. So here, starting with the rational designs, we'll look at the CRISPR-Cas cargos first. So what they do is reduce the offset targeting effects, and this is by modifying the gRNA structures as well as the CRISPR-Cas nicases. And then step 2, we can relieve the immune responses. So this would be alternating Cas nucleases and constructing regulated Cas variants. And then step 3, we can do HDR-induced editing, as well as CRISPR-A or CRISPR-I, which is CRISPR activation or CRISPR inactivation, and this is epigenomic genome engineering. And so this will really provide for improving editing activities, and you can also use base editors. Getting to the delivery systems, we start with biosafety. So you can do the cell targeting capacity as well as bioresponsive materials. For efficiency, we have sufficient capacity, proper physical properties, efficient cellular uptake, nuclear localization, and co-delivery. For targeting ability, you have proper size nanoparticles for biocompatibility, as well as specific particle properties, stimuli responsive materials, and liver targeting ligands. So again, this is really applicable to hepatitis infection and HCC or hepatocellular carcinoma. So this is some great applications of advanced nanotheragnostics for CRISPR-Cas specific to liver infection and cancer. And you can read the Kong paper in Advanced Sciences, which was published in 2021. I want to thank you for your time. These are the current developments and upgrades of CRISPR technology. And I want to thank you for your time.